First of all, I'd like to thank the organizers for inviting EG427 to this very interesting, I must say, event. I'm really excited. I do represent EG427, which is a French company, and you may wonder, wow, far away. I am actually based on the US East Coast, actually in Southern California, and I'm the Chief Medical Officer of EG427. This is the legal disclaimer, which you can read at your leisure. And what is our mission? Our mission is to provide the next generation of precise gene DNA therapy as a local therapy to patients. We are working with non-replicative herpes simplex vectors. Herpes simplex are very unique viruses in the virus world. The majority of all gene therapies are being delivered by adenovirus vectors. Herpes simplex are different. Our first product is called EG110A, and the aim is to silence sensory C fibers. So most of our sensation, like burning, heat, when you eat something, spicy, this is C fiber mediated. Chronic pain is, for example, C fiber mediated. Chronic itch, and so is also the sensation from the bladder. We are planning to initiate a clinical study in early 2024, and we are also looking at a follow-on product, which is called EG132A, which is in preclinical stage for spasticity. Again, these are locally applied gene therapies which are not intended to be systemic, but to really solve a local issue. Again, we were founded in Paris in late 1990. The technology was in-licensed from an academic lab at the Hôpital Cochin in Paris, and we currently have 16 FTEs, including myself, and we have our own research lab with our own ability to generate novel HSV1 vectors. Again, I'm just not going to read all of the basically supporters. They're largely from Europe at this point. This is our basically our management team. Our CEO is Philippe Chambon. Our Chief Operating Officer, Samuel Salo. Our Chief Science Officer is Alberto Epstein, who is one of the leading researchers in herpes virus technology. This is myself. In the pharma industry, I have to disclose, I'm not a neurologist, I'm a urologist, the one with the U, and I've devoted a lot of my life to the study of incontinence, also to neurourology. I come from Allergan, and actually most recently from Eurovant. Then our VP of Research is Greg Sarek. He comes from Roche. And then we have a VP of Strategy, Michael Constantin, who comes notably from the company Renault. This is our scientific advisory board, and I'd like to point out Professor David Fink, who's one of the leading researchers and the first one actually who developed a non-replicative herpes simplex vector for the therapy actually of skin cancers. And so I think we are well set up to proceed. What is a non-replicative HSV1 vector, and why should it be a good idea? So first of all, in contrast to AAV, as I said, adenovirus vectors are the ones, when you read about gene therapy, they're the ones most commonly used. Herpes simplex is relatively new. Non-replicative means it is a herpes virus, but it will not replicate. It will not cause further infection. First of all, it can carry a large load. There is no neutralizing antibody to herpes simplex vectors. It is possible to be treated, and I'll show you an example actually of a recently approved drug, again in dermatology, where the drug was re-administered and re-administered without any immunological reaction. There's no integration into the host DNA, and on the manufacturing side, there's efficient scale-up. And they do have a proven safety record, and I like to point out Crystal Bio, which is a company which focuses on dystrophic epidermolysis bullosa. This is a more rare genetic disease where it basically affects children, starts from early childhood on, where you develop large blistering, huge blisters. And their product, BVEC, can be reapplied and really repairs the underlying mechanism of the skin. This was just FDA approved in May of 2023, which shows you can get non-replicative herpes virus vectors to the FDA and for approval. Now, where does the herpes virus go? There's a national tropism. The herpes virus actually, once you get herpes, knows exactly where it goes. As you know, if you ever had a herpes infection, especially people who had it on their lips, it'll come back. Because once you have the herpes virus, it'll live inside you. It'll live in your nerve cells. And so there's multiple organs, and you can see which basically could be a target for a non-replicative herpes virus vector. And we call it precision because the herpes virus vector knows exactly where it's supposed to go. This is our pipeline. Our first product, that's why I'm here, is EG110A for the treatment of bladder dysfunction. Our first study will be in spinal cord injury patients with urinary incontinence and with the so-called neurogenic bladder, which is called neurogenic detrusor overactivity, which means it is an autonomous, constant. Detrusor is the bladder muscle, for those of you who are not so familiar with it. Constant contraction of the detrusor muscle, which then leads to constant urinary leakage, because urine comes down from the kidneys all the time. You cannot control it. So your bladder fills. And once, of course, there's uncontrolled contractions, you will then start leaking. The second product, as I already said, is in preclinical for spasticity. We're also looking at a potential chronic pain indication. There's undisclosed sensory pathologies and an undisclosed metabolic disease. Again, our lead program is focused on the type C neuron inhibition. This, again, is a replica of a herpes virus vector, and I won't bore you with genetics, gene therapy. I'm also not the expert, I have to admit. Nonetheless, this shows basically it gets integrated, it is under control of a CGRP promoter. Promoter in gene therapy means that is the part that's get integrated into the DNA, which makes the cell produce the gene that it's supposed to produce. That's a promoter. Again, we're looking at type C neuron. They're the smallest nerve fibers. They have very disseminated across many organs, as I already said, heat, burning, cramps, and itching. Our first study is with a local therapy, locally administered into the bladder, similar to Botox. As you may know, Botox also used for overactive bladder and for also neurogenic detrusor activity. Maybe we will in the future also do overactive bladder and interstitial cystitis, a highly underserved, painful condition of the bladder. So there's a large unmet medical need in neurogenic detrusor of activity. Yes, there's oral drugs. They were largely approved for the general overactive bladder. And also Botox was developed. And I was deeply involved in the Botox development for the bladder. However, Botox has two issues. First of all, Botox will, if you still are able to void, and some people are still able to void, it can lead to what is called urinary retention, which means instead of leaking, you cannot go at all anymore, which may be not acceptable, but maybe manageable for people who do self-intermittent catheterization. However, if you're a multiple sclerosis patient or another patient, that is very difficult. The other thing is Botox has to be constantly re-injected. It lasts in the bladder for about 6 to 9 months. It will not last longer. So this is a constant re-injection, and the injection is in a minimally invasive procedure. It's called cystoscopy, where the cystoscope, the scope is inserted, and then Botox is injected. So we're starting off in spinal cord injury, where we believe is the highest unmet medical need for a long-lasting solution. So we're going to study people with NDO. The regulatory side, which was addressed here, there is a pathway to get this approved for NDO because Botox was approved by the FDA and actually approved globally. So there's actually a regulatory pathway. And with the NDO approval, you also will include multiple sclerosis and other conditions like spinal cord or potentially stroke. And then, of course, we will look at other indications, but there is already a regulatory precedent on what you need to do for neurogenic detrusor overactivity. Again, I don't want you to explain to this audience that NDO and spinal cord injury is a high economic burden and a burden on the patient. Nobody talks about the bladder. You know, bladder is not fashionable, I know that. Nonetheless, it is a very important topic and it will really affect your quality of life. So what does it do? The aim is to inject it into the bladder. I hope I get the pointer now, right? Now, where's the pointer? Here, in the middle. So here's the bladder. This is basically a cystoscope, and this is a bladder. Here's the urine comes down from the kidneys here, and it flows constantly. You can't stop it. And here, then the product will be injected here, will travel to what is called the dorsal root ganglia, which are the sensory side, and it is supposed to rest there. It will basically deliver the light chain of botulinum toxin F, which is the sensory part of a botulinum toxin, and inhibit the signaling, the reflex signaling back to the bladder, and therefore reduce the bladder contractions. This is what we're aiming at. And we can also show you images in rats, we are not yet in humans, where you actually show that the product does travel from here to there. So here's a rat model. You can see here it was here injected in the detrusor and sort of traveled here. And this is an image. It's called RNA scope. It's an imaging. You can image these viral vectors on where they are. And this is an image of the product shown by RNA scope, the red dots, if you will, in the dorsal root ganglia of the rats, which means it does travel indeed, as it should, from the bladder along these C fibers to the spinal cord. Again, a little bit about the action of botulinum toxins. This is a general depiction of what a botulinum toxin does. Botulinum toxin actually binds to something called a snare complex. Well, this is the normal normal electrophysiology with acetylcholine. It binds and then it releases the neurotransmitter. So Botox works on acetylcholine. Some of you may remember acetylcholine from your medical school. What EG110A does, it will also bind to the snare complex, and here's the light chain of botulinum toxin F, but it will not bind here, it will not release it, and it will not release the selective sensory transmitter, which is called VAMP. So this is what it will do with a light chain of botulinum toxin F. Botulinum toxin F and the light chain is specific for these type C neurons. We did do a rat study in actually a general bladder model of bladder overactivity, which is called a capsaicin model. Capsaicin is a highly irritant substance, and it was infused in rats. And you can see here versus control, EG110A administered via injections was able to reduce these bladder contractions. The rats developed well, and what also is very important, it did not inhibit voiding function of these rats. So, what is our clinical trial plan? It's a phase 1b/2a. It's in spinal cord injury patients. We're currently planning all levels because the NDO does not depend on level of spinal cord injury. You have NDO or you do not have NDO. It's an open label dose escalation study. We're thinking of 16 patients. We're going to observe them for 52 weeks. We're going to do a pre-planned interim analysis at week 12. Week 12 is the accepted regulatory endpoint in most urology, functional urology studies, including an NDO. And as gene therapies require very long-term follow-up, current FDA guidance for herpes simplex vectors is 15 years. We're going to talk to the agency 5 years. And the idea is to have an extremely long duration of action. We don't know how long, but very long. Certainly far longer than Botox, trying to avoid as much re-injections as possible. And we will see. Again, the next steps, we had a positive pre-IND meeting with the FDA. We also spoke to the UK authorities called MHRA. We know what to do preclinically. The clinical lots are being manufactured under GMP. We heard before how important the GMP manufacturing is. And we have a clinical plan drafted, and we're also currently evaluating EG110A in pain models, again in rats. The secondary program is in spasticity, which is basically far more in earlier stages. Spasticity, as you know, is basically an excitatory. The muscle cramps is an uncontrolled signal, and the muscles cramp. So there, of course, Botox is also being injected in spasticity, but there, for some reason, the striated muscle has a shorter duration than the smooth muscle. In bladder, it's every 6 to 9 months in the striated muscle. As for spasticity, it's every 3 months, where you need re-injections. And they're implantable pumps, baclofen, and our plan is to basically, again, provide a long-term solution. The plan is, again, to use the tropism of the herpes virus, and it will be injected into the affected muscle. It'll travel again as to the dorsal root ganglia. In this case, it's not going to work on the C fibers, but it's going to basically transform an excitatory neurotransmitter into an inhibitory neurotransmitter, which means it will inhibit these uncontrolled muscle contractions. The vector landscape is actually quite interesting. So here are all sorts of oncolytic viruses already being developed. The non-replicative vectors, this is Crystal Bio, as I said. This epidermolysis bullosa is the first approval of a therapy for a treatment which had no prior therapy, and also shows that the herpes, non-replicative herpes simplex vectors can be delivered repeatedly and also safely. That is very important, without any systemic immune reaction. And then there's other developments, but Crystal Bio's approval certainly was a landmark in the field for non-replicative herpes vectors. EG427 is the most advanced company using non-replicative HSV-1 vectors in neurology or neurourology. Crystal Bio is focused on other indications. So in summary, a herpes simplex vector can carry a long payload. As you remember that famous balloon, the weather balloon, the payload, that also applies in vector therapy. How much can they carry? How much genetic information can they carry? It'll be able to be redosed. There's a documented safety with another vector, and we believe that it's efficient manufacturing. Manufacturing for a replicative, non-replicative herpes vector is far more cost efficient than for any adenovirus vector. It will be, if it works, it'll be a therapy which is highly targeted towards exactly the site of action, non-systemic, and we really believe it'll have a long-term expression of the, in this case 110A, of the light chain of botulinum toxin F. And we do have internal capabilities to further develop other vectors for other indications, and we also have a broad platform to allow for multiple partnering. So thank you very much for your attention and for inviting us.